If you want to figure out if a disease is linked to a particular pathogen, you have to study both the healthy organisms and compare them to the sick organisms. And so for the first postulate, as I said, the microbe associated or thought to be associated with the disease caused in the unhealthy host must not be present in the healthy animal or host. So that's postulate number 1. A particular microbe must not be found in all cases of the disease and must not be present in healthy animals or humans. The second postulate is that the microbe must be isolated from the diseased animal or diseased human, and then a researcher has to produce it in a pure culture in the laboratory. So you have a diseased animal, you have a diseased patient, isolate the microbe, and then produce it in a pure culture. That means that you're going to have one single organism in that culture, the isolate that you extracted from the diseased patient. Now, once you take that culture and you grow it in a pure form, then you want to test it by injecting some of that culture into a healthy, susceptible lab animal. So when this happens, this lab animal that was once healthy should then be able to produce the same disease from which was produced in the unhealthy organism where the microbial isolate came from. So the third postulate states the same disease must be produced when microbes from the pure culture are inoculated into a healthy, susceptible laboratory animal. Well, that makes sense. That gives us control and experimental value to compare with, so that way you can see if the healthy, susceptible animal now has the same illness that the first unhealthy test subject had. The fourth postulate of Robert Koch's is that the same microbe must also be able to be recovered from the experimentally infected animal, and it must be grown again in a pure culture. So once this animal starts reflecting symptoms of the same disease as the host organisms from which the isolate stemmed from, that microbe must be recovered or observed in the infected animal, and it needs to be extracted from the newly infected animal and then reproduced in a pure culture. So if all of these postulates are met, what we will find is that sick patients or sick test subjects only come from the injection of a microorganism into their what was used to be a healthy system. So this is demonstrative of the evidence that was used to refute the theory of abiogenesis, meaning that life just doesn't spring from anywhere. It is not spontaneously generated. It comes from microorganisms. Disease is brought on by microorganisms. It's not spontaneously generated. So that's what these particular postulates were able to demonstrate. Now, some other features or attributes with Koch's postulates are the ideas that if an organism fulfills Koch's postulates, it has been proven to be the cause of that particular infectious disease. So, linking the causative agent with the disease is the primary basis of Koch's postulate. This, as I said, ended up proving the germ theory of disease, which states that diseases are caused by germs or microorganisms. And then because he was able to demonstrate this, all of the techniques that he used really provided a great foundation for establishing a lot of the laboratory techniques that exist today. So culturing and the identification of microorganisms became a very critical, very critical strategy that was employed in laboratories in order to start investigating diseases and ways that we can treat diseases. As doctors, chemists, research scientists, this is what they wanted to figure out. How can we save lives? You know, what's causing death? These are questions that to us seem kind of second nature because, you know, of all of the advances and knowledge that we were brought into this world knowing at our particular generation time. But back then they didn't know all of this stuff. So there's still a lot of room for exploration. Now, even though we know today that a number of these postulates are pretty accurate, there are still some instances in which those postulates haven't been able to be proven or are not able to be fit. One thing to think about why Koch's postulates wouldn't be able to work properly would be the idea that the bacteria that is isolated from one diseased host may not be able to thrive in a new diseased host in which it's placed into. Or it could be the disease isn't even caused by bacteria. If it's not brought on by bacteria, then the disease can't replicate inside of the new host, which it's injected with. So in a sense, the transfer of injection doesn't work properly. Or some organisms can't be cultivated as easily as you would think. So there's some circumstances in which the postulates, you know, they can't be fulfilled. So lastly, I just kind of want to touch on, you know, some different careers in microbiology. Remember, this is an introduction that we're doing our first chapter. So hopefully you're gaining some insight into the things that have constructed this field. We've talked about the history. We've talked about the significance. We've also talked about, you know, the roles that microbes play. But now we want to kind of, you know, just highlight at the end of our chapter different careers that exist out there. Obviously, individuals like myself who love microbes and love studying them, love researching, love talking and teaching about them, we're referred to as microbiologists, because micro means small. Biologist means, you know, the study of life or one who studies life. So a person who's a microbiologist, you like to study small things that are living primarily. So there's a whole lot of different fields that are in this. There's bacteriology, there's phycology, protozoology, mycology, parasitology, virology. Bacteriology is strictly the study of bacteria. So you don't deal with fungus, you don't deal with algae, you just focus primarily on bacteria. Phycology is the study of algae, which is very, very interesting, especially if, you know, they can figure out how to help start our cars based on the use of algae or biofuels instead of our current dependency on oil. We also have the field of protozoology, which is an area where you are studying the group known as the protozoans. And these are the small animalcules that Antoine von Leeuwenhoek was able to observe under the microscope primarily. Some of them have flagella, some of them look very alien-like, they have little hairs around them, they have weird different types of structures used to ingest food. So all kinds of really neat stuff in the field of protozoology. And there's an incredible amount of diversity that exists in that area. And then there's mycology, which is the study of fungus. There's basidiomycetes, ascomycetes, glomeromycetes, and zygote fungi, all kinds of different fungus that exist really, and they all have very interesting attributes to them. If that's something that you're interested in, I would definitely recommend taking a field course where you can just observe all the different diverse fungus that's out there. Parasitology is just the study of parasites. This is also very fascinating. You observe host-parasite interactions, and that can range from a parasite using a host as a platform to absorb nutrients, or it could be used, that host could be used as a platform or environment where the parasite will, reproduce inside of it or lay eggs inside of that host, using the environment, again, as nutrients, but also as a foundation for a nesting kind of site. There's all kinds of parasites that are out there, and they all can be, many of them can be, mostly parasites. With parasitology, you're studying organisms that don't benefit another organism. They instead, they feed off of them in some form or fashion. And then virology is simply the study of viruses. All of these fields are incredibly important to us because they help us to understand organisms that are pathogenic to us and help us to develop ways to treat as well as live, in cohabitation with these organisms that are all over in our environment. Medical, so you can have all kinds of different scientists in these fields. A bacteriologist would be in bacteriology, a phycologist in phycology, virologist in virology, mycologist for mycology, etc. In the medical microbiology, this is among some of the most important, this has some of the most important careers because we are talking about saving lives. You study pathogens as a researcher, as a scientist. You link those pathogens to associate them with different diseases that can occur in the body. Or you watch those types of interactions and figure out different therapies and treatments to hopefully save lives, or save an individual, from suffering from that disease. So there's all kinds of great fields. Pathologists, they study how disease works. Medical technologists, they'll extract samples from patients. Clinical biologists, they also extract samples from patients, and then they just culture and identify those organisms in the laboratory all day long, or in some cases those viruses, and that assists with diagnosis, and that also assists with helping doctors to figure out proper treatments to use. So they play these types of fields. We have a lot of careers in them which play really big roles in helping to save lives. Epidemiology is also another kind of study that falls under microbiology. Individuals that study epidemiology are known as epidemiologists, and these individuals, they are not very lab savvy. They're more so they're more kind of the social sciences side, I think, of microbiology, where they start watching how pathogens are transmitted in populations, how do epidemics occur, and then they implement. Individuals in this field who have careers in the field of epidemiology are known as, I think I said already, epidemiologists, but they employ measures to hopefully prevent diseases from happening. So outbreaks of STDs, outbreaks of children being infected with enteric bacteria, which are bacteria that can cause gut inflammation and just make individuals very sick through ingestion. So something like a salmonella outbreak. They also observe aseptic techniques that are occurring in different laboratory settings, how they study how infectious diseases are treated. They study immunology, can produce, they also study the production of vaccines as well as the administration of vaccines. And so all of these components are tied to the field of epidemiology, and there's just a variety of careers that are out there that can assist in us monitoring and surveying our populations for epidemic outbreaks and to figure out how to control those, which is incredibly important. So that's the end of our first chapter 1. So I hope you learned a lot, and I hope it gave you kind of an insight into how this class will be run. So each week I'll post 2 lectures, and then you guys will be able to hopefully listen to me as I go through these PowerPoints. So let me know if you have any questions and send me an email. All right, have a good day.